Dear readers, it is a great honor to share our work with you. Today, the topic we share is modulation of rhizosphere microbiota by Bacillus subtilis R31 enhances long-term suppression of banana Fusarium wilt. Fusarium wilt is a devastating disease in banana plantations worldwide. By control bacterium Bacillus subtilis R31 is likely to prevent and control banana Fusarium wilt through regulating the structure and function of banana rhizosphere microorganism flora, and help potential biocontrol bacteria grow into the plant roots. We observed a convergent secondary succession of the rhizosphere bacterial community during naturally suppressive soil and R31-induced suppressive soil. The highlights of this article include R31's modulation of the rhizosphere endures even after its disappearance. R31 modulates core rhizosphere microbial genera and functional pathways related to disease suppression. R31 effectively controls banana Fusarium wilt by restructuring the rhizosphere microbial community. Endophytic bacteria in roots demonstrate greater effectiveness against Fusarium wilt, emphasizing the importance of strain interactions for biocontrol. We performed 16S rDNA amplicon sequencing to document the changes in rhizosphere bacterial communities of R31 treated and control samples in years 2019 and 2020. There was no significant difference in alpha diversity between R31 treated and control groups. The distribution of the samples is not significantly different between R31 treated and control groups. R31 was no longer present in the banana rhizosphere 1 year after application. The relative abundance of these 6 genera did not significantly change between 2019 and 2020 in either group, except for Bacillus in the control group. The emergence of R31 biocontrol bacteria affected the succession of suppressive soil. A similar phenotypic prediction and phenotypic contributed to the suppressive soil and R31 induced. The numbers of isolates retrieved from HR9, He7, and HS7 were comparable to those from SR4, SE9, and SS10, respectively. The strains with close phylogenetic distance to R31 had weak beta-glucosidase production, strong protease production, weak catenase production, and average cellulase production. Strain SR50 also showed antagonistic activities against the FOC004 and FOC009. The antagonistic activity is more closely related to the enzyme production activity of the strains than to their phylogenetic distances. The Halo bioassay showed that R31 GFP showed boundary phenomenon with 13 strains. In the biofilm mixing experiment, R31 GFP coexisted with multiple strains, and the survival of R31 GFP was different under different mixing ratios. In the co-culture system, some strains coexisted with R31 GFP, while others did not survive. The antagonistic tests showed that R31 had no significant effect on the inoculation distance of the tested strains. The results of pod experiment show that the bacteria isolated from the inside of the root system R31 application shows better integrated control effect compared to those from the rhizosphere R31 application. These results show that the control effect of R31-induced soil bacteria is better than that of naturally suppressive soil bacteria. In this study, we found that the applying of R31 can provide the convergent secondary succession of the rhizosphere bacterial community in naturally suppressive soil and R31-induced suppressive soil. The common disease-suppressing soil induced by R31 can be transferred to specialized types of soil disease inhibition.